Hi guys, and welcome to another video. Today I'll be doing a draw my life on how I went from a diploid cell to a haploid cell. I'm talking about the process called meiosis. This process produces gametes, or sex cells, with half the number of chromosomes as a parent cell. My life begins in interphase. The cell must go through interphase before going through meiosis 1. Cell growth occurs during the G1 phase. Chromosomes are replicated during S phase. Finally, in G2 phase, the cell prepares for division by forming structures related to meiosis. After interphase was complete, meiosis 1 could begin. There are 4 stages to meiosis 1: prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 1 turns one diploid cell with 46 chromosomes into 2 haploid daughter cells with 23 chromosomes. Going through prophase 1 is pretty interesting. The centrioles start moving towards opposite poles of the cell, and spindle fibers begin to form. My homologous chromosomes line up side by side. This is called synapsis. These tetrads, one from each parent, then exchange genetic material, crossing over, that provides genetic diversity in this phase. Also in this phase, the nuclear envelope begins to break down around the chromosomes, and the centrioles should almost be at the opposite poles of the cell. It was now time for me to experience metaphase 1. The homologous chromosomes have lined up at the equatorial plate. In their pairs, one homologous chromosome will be on either side of the plate. The chromosomes align themselves randomly, so for example, the blue maternal chromosome could be on the same side, or on this case, different sides of the plate. Anaphase 1 begins with the chromosomes separating and moving to opposite poles of the cell. Remember, the sister chromatids are still together and the centromeres have not split. During anaphase 1, if the homologous chromosome pairs do not separate into opposite poles, both sister chromatids are pulled to the same side of the cell. This is called non-disjunction. It produces gametes that have too little or too many chromosomes, and can affect meiosis too. In telophase 1, my life starts to get really exciting. My chromosomes uncoil in the nuclear envelope that starts to reform. The spindle fibers begin to disappear. The cells begin to pinch in the middle, creating a cleavage furrow that divides the cytoplasm until cytokinesis occurs. And wow, 2 haploid cells are formed. In meiosis II, the process is very similar to mitosis, the asexual reproduction of a diploid cell. Prophase, metaphase, anaphase, and telophase also occur in mitosis. Prophase II begins with the chromatin condensing into chromosomes that consist of 2 chromatids held together by the centromere. The centrioles move to opposite ends of the poles, and the spindle fibers begin to reform. The nuclear membrane also disappears. This process occurs in both cells created in meiosis I. Now it's time for metaphase II. In metaphase II, the haploid number of chromosomes line up at the equator of the cell, and the spindle fibers attach themselves to the centromeres of the chromosomes just as they would in mitosis. Anaphase II begins with the spindle fibers pulling the sister chromatids to opposite poles of the cell when the centromeres divide, just as they do in mitosis. Unlike in mitosis, these chromatids are not identical. During anaphase II, non-disjunction may occur again. After a normal meiosis I, non-disjunction during anaphase II can lead to 2 gametes having the usual number of chromosomes, while the other haploid cell produced gametes with 1 fewer chromosome and 1 extra chromosome. Gametes that have an extra chromosome due to non-disjunction can lead to the genetic disorder Down syndrome, in which the person can have an extra piece of chromosome 21. It's finally time for the last phase in mitosis II, telophase II. The chromosomes are once again at separate poles of the cell, and the nuclear membrane reforms, forming 2 distinct nuclei in each cell, and the cleavage furrow develops again. Cytokinesis occurs at the end of telophase II, and 4 genetically different haploid cells are formed, each with half the number of chromosomes found in the original diploid cell. And that's it. That's my life so far. Through the full process of meiosis, my DNA has started from a single diploid cell, and now I am one of 4 haploid cells. I hope you learned something new about meiosis and how haploid cells are formed. Bye for now. See you next time.